Bismillah ar-Rahman ar-Rahim. My dear fellows and students and colleagues and postgraduates and those who are listening or want to listen, the art of medicine, how we reach to the beneficiary, that is the patient, how we can help the patient. And this is the background knowledge you have to achieve. These are the revisions which you have already done in the first professional MBBS or second professional MBBS. We are telling you something about infection and the disease process, infection leading to the diseases. Or we can say the infective diseases, infectious diseases. Means the disease can be spread to the one object or subject to the other subject. Now it is the specialty which is called infectiology. And these are the 2 or 3 lectures which I have taken actually. These are base what we will discuss. I think it will be around 30 or 35 lectures or tutorials regarding this infection and the disease. You are proud of one thing, remember, of your what is called allopathic medicine, that you can evaluate the disease and eradicate the disease 100%. There is no doubt about it. That is why the allopathy is so much popular. At one time, some diseases were not cured. The patient died. And one of the disease that is called is the plague. Plague, that is. It is called black fever. Plague is the oldest disease known in the history. And then coming the sexually transmitted diseases like syphilis in Europe and in America and many parts of the world. And this disease has killed a lot of populations. And one time there was no specific therapy. The revolution in the medicine emerged when the light microscope has developed, and then the large microscopes have developed, magnifying the images, and then electron microscopy. On the one hand, on the second hand, the therapeutics is concerned, the drug which was first invented, first antimicrobial actually we can say, which was invented by Fleming, and then he got Nobel Prize for it. And they said it was cure for every disease because it controlled the syphilis in that zone, in France, in Germany, and other, because the scientists of that zone. And the king has crowned him also. And he said that you are a better person than me because I cannot, control the process which is going on, and people were dying at that time. And he was the luckiest person at that time and will be honored by Nobel Prize. Afterward, a lot of antibiotics have arrived, and though a lot of advances have been done. Maybe it is a microbiological concept of any disease. And there is a theory which is going on, that every disease has some of the link among the microbes, because the microbes or bacteria, for example, they are the oldest I will say, ancient living organism. When may Almighty Allah detached the earth or created the earth. The solar system has been regulated at that time. So this is a story. Now, I am Professor Syed Ali Haider. I am visiting professor of medicine, GSMU. You have to understand these terminologies. They are microorganisms, infective to the humans. Because the microbiology is concerning, it can affect the the subjects or the objects. Everything is covered with the microbes all over the world. No single part of the earth, whether it is on the surface or above the surface, we do not contain these microbes, microorganisms. But everybody is living fine. You are enjoying your life. Because the human actually developed the immune mechanism. Every living organism developed immune mechanism or defense mechanism against these microbes. This is the war which is going on between the microbes and the immune mechanism which has developed in the persons. Remember, this is the war which is going on. But actually the underlying knowledge, or the basic knowledge, or the fundamentals of the microbiology should be known to the students when they are appearing in the examination. For example, examiner asks, can you tell me some of the diseases which are known to cause from gram-positive bacillus? My student answered it like this: yes, sir, very common diseases, salmonella organisms, they cause disease, very common disease. On the other hand, another student has been asked like this: gram-positive, gram-negative organism, do you know some of them? He said, yes, sir. Antimicrobial stolytica, Giardia lumlia. If you were examined at that time, will you like to pass this student who will be then become after one the medical providers or the health provider afterwards? No. Why? Because the basic knowledge is missing. If you don't know it is salmonella, or you don't know about Escherichia coli, you don't know about the Antimicrobial stolytica, you cannot understand this infectiology. So, now these microbes are many fold. Though we have actually microbes which are interested in the human microbiology are bacteria, Schindleria, mycoplasmas, rickettsia, parasites, viruses, fungi, parasites, rounds, virions, viroids. Now, some of these terminologies are not known to the students at one time. Now the people are, via internet or via the, news, they know about these things. Lyme disease was a, nightmare in some part of the world at one time. They said it is a nightmare. It is a nightmare. The horror movie scenes related with the Lyme disease, that the patient had problems and something happened to this person. And he was occupied by some ghost or by a spirit, etc., etc. These were concepts at one time. Now Lyme disease, you know that it is caused by a spirochete. Every disease can be related to the microbe at one time. It is 2023. We don't know after 2020, 50, rather after 25 or 30 years, probably for example, the peripheral vascular diseases, the ischemic heart diseases, or the cerebral accidents, which is a common problem, or relating with the oncology, related with the hepatology, related with the fat, the errors in the fat metabolism, etc., can be related or can be found to be, in fact, related to the microbes. We don't know. So these are the water bacteria, Schirmaria, Microbiology, Rickettsial, these are the microbes of interest. Bacteria are ubiquitous, mostly free living organism, often consisting of one biological cell. So it has got plasmalemma, it has got some nuclei, it's got the the cytoplasm and the organelles of the cytoplasm. They constitute a large domain of the prokaryotic microorganisms. Large domain. Those prokaryotic microorganisms mean that they have got a definite system of metabolism in the body. Just like you are taking oxygen and you are expiring carbon dioxide, and there is a cycle of citric acid is going on the beta oxidation is going on, etc., and the end products are coming. You are creating biomass, etc. Now these microorganisms are large domain of bacteria, typically a few microcentimeter in length. Bacteria were among the first life forms to appear on the earth and are present in the most of its habitat. Habitat means that where it occurs. Now, classification of the bacteria, let's see the bacteria on the basis of the qualitative analysis. There may be commensals, there may be symbiotic bacteria, there may be pathogenic or invasive bacteria. Now, commensals are those type of the microbes that reside on the either surface of the body or in the mucosa. Remember, they are without harming the human health. The microbes living in the harmony with the human mostly consist of the bacteria, also known as commensal bacteria, which are 10 times more than the cell present in our body. There are a lot of commensal bacteria. Specific to the site, they are important. For example, let me give you an example about the Bacillus catarrhalis. Is commensal to the anterior nares, but it is not commensal to the bronchi. It is not commensal. For example, Escherichia coli is one part of the coliform bacteria which is present in your body and is not harming to you. Neither it is harming nor it is benefiting. A specific place is there for Escherichia coli. But if you put the Escherichia coli in the urinary tract, it will cause problem. It is not commensal there. That is why the Staphylococcus epidermidis is commensal on your skin, but not in the other part of the body. It will cause problem. These are the examples of the commensals. Commensals have got a specific site. Every part of the body in your body, there are a lot of bacteria which are present 10 times more than the cell present in our body. So much because their methodic divisions are very, very quick. The replication power is much. Now, symbiotic bacteria are those bacteria which are actually present in your body, but they benefit you. Symbiotic bacteria are bacteria living in a symbiosis with another organism. So they work in a group or each other, helping each other. For example, rhizobia living in the root nodules of the legumes provide nitrogen fixing activity of these plants. Symbiotic bacteria are present in you, like Escherichia coli in the small intestine is beneficial because it will give you vitamin K to your body. Without you are taking from the external source, the body itself providing you vitamin K, which is this water soluble, the lipid soluble vitamin, and very important for the extrinsic pathway of the coagulant factors of the liver. This is one example of symbiosis. There are a lot of examples in your body. These are beneficials. These organisms are beneficials to the body because they are providing you the different substances, or they are important because they will not tolerate the other bacteria which are coming inside the, for example, in colon or in intestine, etc. That's why if you give the antibiotics, all bacteria will be killed, the symbiotic bacteria will also be killed, the commensal will be gone, and if you, the invasive bacteria enter afterwards, just afterwards, this will cause a problem in the intestine. There will be severe type of the enteritis or gastroenteritis or gastroenterocolitis. That is why the antibiotic, if you are taking antibiotics, it is old saying that you should supplement with multivitamin because the vitamin will not be generated now. And bacteria which are present as symbiotic, they were providing some of the vitamin like vitamin K, for example. And secondly, the fighting force is gone. So these invasive bacteria which are coming after the eradication of the symbiotic bacteria will cause the disease. And one extreme example of it is the antibiotic-associated diarrhea or Clostridium difficile-associated diarrhea. It's one of the pathophysiology of the Clostridium difficile or antibiotic-associated diarrhea. Another type of the organism is the virulent invasive bacteria, pathogenic bacteria. Now they are infective, they depend upon the infective dose, their ability to enter into, replicate within very, very quickly and persist at the host site that are inaccessible to the commensal species. The commensals are not present at that. Commensal species are specific to the sites. Symbiotic bacteria are not there. They cannot fight. They are defense mechanism. Rather, I would say this is the first defense mechanism. It is not there. And they penetrate into the body and will cause the disease after the incubation period, when there will be replication, so much so that the third line of defense or the fourth line of defense will not be able to counteract this problem, then an infective disease occur. This is the end of the incubation period. That is the incubation period when the microbes enter into the body and there are clinical symptomatology. This is called incubation period. Now, classification according to the structure is concerned, there will be structurally they are classified like cocci or bacilli or corynebacterial, spirals or mycobacteria, etc., etc. Or branching or microorganisms like fungi. Now, classification according to the stain, they classify as gram positive or gram negative or acid fast, and other stains also are there. Leishman stains and the methylene blue stains and other specific stains are there to identify the different type of the microbes. Usually the gram positive, the gram negative, acid fast. These are three common. The gram positive cocci. Cocci is always, they are acting in chains, bundle chains. They are rounded in shapes. If they are stained by this, at one time we are doing it, the gram staining. The gram staining, it is just like this. It's positive stain there. So example are staphylococci Streptococci, pneumococci. Streptococci, lot of disease. We will discuss on the next issue. Streptococci or pneumococci. Take one by one all diseases. Do not say that it is uncommon, I will not study. For example, my friend was failed because he did not know in SCP examination. He did not know about anything about the Legionnaires' disease. They failed him because it was a differential diagnosis of atypical pneumonia. And the subject, the long case, there was a case of pneumonia. The thing is this, the person was denied that he was never heard in his life about the Legionnaires' disease. How can he will be passed? Becoming assistant professor afterwards in 1 year or 2 years, then associate after 5 years, then after 10 years professor of medicine. And then he is the king of the unit, that's all. And he will be sitting in the room, not abandoned for the other people to enter. So it means that these things are important here to understand how. Give me the example of gram-positive cocci, streptococci, pneumococci. Give me the example of the gram-negative cocci. Negative means that it will not stain this grams. It is like this, pale, purple it is not red. Meningococci and gonococci. There are 5 types of cocci. 3 are positive and 2 are negative cocci for the ground staining. In this fashion we are dividing it. Gram-positive bacilli, although they are very few in number. Why that person was caught actually. My student was caught because he has not said that anything gram-positive bacilli. He said gram-positive bacilli salmonella. Salmonella is not gram-positive. And bacillus anthrax, a good example, is gram-positive. There are other examples as well. About the Corynebacterium, yes. Leptospira monocytogenesis weak, yes, the weak gram-positive bacillus it is. And others also, few others which are not important to understand now, to name them. Gram-negative bacilli are multiple. The people are, the clinician or attending health carers, they are terrified from this type of infection. It is highly infective organisms. Salmonellas, Escherichia coli, Proteus, Shigella, Pseudomonas, and the Bacillus catarrhalis, Klebsiella pneumoniae, etc. A very, very long list is there, of course, in this case. They are gram-negative bacilli. A structural change of this, some of them, it is the cornification of the bacterium, club-shaped. Corynebacterium, for example, diphtheria and there are other corynebacteria as commensals are also present in your body. So they generate their name as club-shaped, but they stain with the gram as positive. Corynebacterium. Although it seems like gram-negative, when they stain the gram-negative. Mycobacterium, weakly gram-positive, although specific stains are used. Acid-fast, that is why they are called acid-fast bacilli. They grow on the specific media only. And there are about 90 species in this phylum. Actini, Mycetota. And one of it is Mycobacteria, large bacteria in size. And they are specific stains which we use Richmond and other stains. Gram staining are weakly positive, specific stain for acid fast are there. and they classify them that this is the stain which you are using, acid 5 bacilli are present in sputum. So diagnosis will be mycobacteria. Which mycobacteria? The commonest one is the Mycobacterium tuberculosis, but there are a lot of mycobacteria also cause disease. When we will discuss the immunosuppression like HIV, we will discuss. Now spirochetes. Spirochetes, any of the group of the spiral shaped bacteria they are. Like this. It's like a worm. Humans causing disease is syphilis. This is very important disease, I said at one time. Weekly gram positive it is. And yaws and Lyme disease and relapsing fever. Example of the general of the spirochetes include the spirocheta, Trypanosomas, Borrelia, which all are important. All are important for the human. Borrelia for the Lyme disease. Leptospira hemorrhagica, it's important disease. Trypanosoma causing the syphilis and spirochete, which are causing the red bite fevers and other fevers as well. Schirmadias, Schirmadia pneumoniae, Schirmadia trachomatis, Schirmadia psittaci. I have seen all these. Pneumonia, yes, atypical pneumonia, cause of atypical pneumonia. Psittaci, cause of the bronchiolitis, a cause of the lower respiratory tract. Schirmadia trachomatis is a very common organism for the eyes to cause blindness. If it is in the genital tract, urinary tract, it is one of the commonest causes in the world for the urinary tract infection. And it is definitely nitrite negative type of the infection. The Schirmadial cell has a gram-negative cell wall structure, and it consists of the outer membrane, Schirmadia, and the inner cytoplasmic membrane. There is no double membrane here, one membrane. That is why the Schirmadia, in containing the penicillin binding proteins, Schirmadia-like measurable muramic acid, which is the cause of the fever in most of the cases of the other organism as well, but in a very lesser amount. Measurable, Schirmadia-like measurable muramic acid. You cannot measure it because the quantity is much less. A structure found exclusively in the peptidoglycan. There will be vacuolated structures, elementary bodies, cell nucleus is there. The membrane is, in this case, is plasmalemma and cytoplasmic membrane. Not the membrane you have studied in your body. It is a different type of the wall membrane. The Microorganisms. Microorganisms are the genus of the bacteria that, like the other members of the class Mollicutis, lack a wall. It is devoid of wall around their cell membrane. Deficient walls. Spore, like this. Irregularity is there in the walls, and the peptidoglycan is absent in this case. Therefore, the measurable amount of the muramic acid will not be there. This characteristic makes them naturally resistant to antibiotics, because most of the antibiotics act on the cell wall. The target cell within the synthesis, they disturb the synthesis of the cell membrane, targeted the cell membrane. But they say that the cell membrane is not present, or if it is present, it is irregular, it is not complete membrane. So they are very, very difficult to manage. They can be parasitic or saprophytic. It may be dependent on your body, it may be saprophytic, topic to the other sides, on the sand, on the trees, or other things. Saprophytes. Mycoplasma pneumoniae is one organism which is important. And remember, this organism may be misleading, not knowing you the person, which will cause the syndromic type of a problem. I will inshallah discuss in a few weeks. Rickettsias. What are rickettsias? The rickettsias are the diverse collection of the obligatory intracellular, they are intracellular, gram-negative bacteria, which are carried inside by ticks. Even zoonosis is there. Depending on the ticks, depending on the lice, fleas, mites, chiggers, and mammoths. So it is zoonotic disease. They are spreading via animals. They include the genera Rickettsia, Ehrlichia, Orienta, and Coccidia burnetii, for example. Yellow fever. These zoonotic pathogens cause infections that disseminate in the blood to many organs. So zoonotic, if there is somebody who will be talking to you in the future about zoonotic pathogens, you must tell this also. Rickettsias. Because they brought into the body from ticks or lice or fleas or mites, chiggers. Malaria is another example, for example. And my dear disease, that is dengue in our country, is zoonotic disease. Fungi. Fungi is a type of the eukaryotic organism. This is not organ different than that. It is not prokaryotic. They have got the definite metabolism other than the prokaryotic, belonging to the kingdom fungi. They branch themselves like plants. They are between the animal and the plant. Plants, animal, protozoa, monera. So they are branching also, they stain also, there are definite mechanisms of metabolism also, and their cell membranes are very different. Commonly encountered forms including the yeast and the mold and the truffles and the mushrooms. These are fungi. The human infested fungi, there are two types of the fungi. One are surface fungi and one are deep fungi, mycosis. The subject is mycology. Even the mycology is now the subspecialty of the clinical microbiology. Some are very, very higher persons who are expert in the fungi. The human parasite, dependent on the person, the parasite are dependent on human. Protozoa or multizoa. Multizoa means there will be cystodes, there will be other one, the nematodes, etc., etc. Inside the body, an active parasite are those which are superficially affecting on the skin. So they are divided into endoparasite, and the ectoparasite, endoparasite will divide again into the different phylum, and phylum will then divide into the families, the genera, etc. Prawns. A prawn is a misfolded protein that can transmit its misfolded shape onto the normal variant of the same protein. Prawns are the causative agent of several transmissible and the fatal neurodegenerative diseases. Creutzfeldt-Jakob disease, for example, one disease, but there are other diseases as well. वो कहते थे ना वो बर्ड फ्लू फीवर हो गया किसी को, और फला हो गया और ये हो गया. प्राउन डिजीज है ये. अभी जो ये कोरोना वायरस के बारे में भी ये कहते हैं, ये एक्चुअली जो है ना ये इतनी ज़्यादा म्यूटेटेड है, दैट इज़ उसके जो है ना वो कल्चर करना वेरी डिफ़िकल्ट है, और उसकी सेल वॉल को बताना बड़ा मुश्किल है. अलबत्ता उसके आप इन्फेक्टिव प्रोटीन पकड़ सकते हैं. तो वो अभी लटक रही है कि ये एक्चुअली वायरस है या प्राउन है. प्राउंस, वाराउंस, ये सारे जो डिपेंडेंट है वायरस आर द ऑर्गेनिज्म मेंबर which cannot be seen with the large microscope. With a higher microscope, you can indirectly see, but with the electron microscope, you can be accessible to them. Other than the virus is the terminology we use usually. But prawns, the virus means complete. It's got cell wall, it has got the system, it is a living subject like you. But prawn is concerned, or the variants are concerned, or varioids, these are the derivatives of the or the lower groups are coming, which are not accessible to the human now. The virions, that is complete infectious virus particle, includes genome comprising one of the few molecules of either DNA or RNA. They consist of the DNA or RNA surrounded by the morphologically defined protein coat. There is a protein coat, the capsid. The capsid and the enclosed nucleic acid together constitute the nucleocapsid. Here the capsid, this is seen here, there is the DNA inside and there is the RNA here. And the cell wall is not complete, there is specific structure in the cell wall, tegument, and the envelope antigens and there are other glycoproteins. They are specific diseases related with the virions. We'll see when we discuss. Viroids, again only, they are infective proteins, I will say. Whereas small single-stranded circular RNAs, they are infectious pathogens. Unlike viruses, they have no protein coating at all. All known viroids are inhabitants of the angiosperms and most cause diseases, whose respective economic importance in humans varies widely because viroid diseases are misnomer because we don't know much about the viroids are concerned. They are infective proteins, only RNA is there, no DNA, no wall, no capsid. Lowest modes in an organism can be accessible by the high-powered electron microscopy is viroid. It is a very interesting subject that lesser than this, smallest may be possible. We don't know in the future. Probably some of the particles are present which are lesser than viroids, very small. That is why I said that every disease may be related in the future to some microorganism. So I thank you about it. And in the last but not the least, remember at one time the acid peptic disease was a disease. They said that this is a disease of the deficiency of the mucosal surfaces and the increment of the acid. So the war is going on between the defense mechanism of the stomach or duodenum mucosa and the hydrochloric acid and the pepsin which is acting on it. The war between this. You are balancing this is okay, fine. If one is superseded, then the acid peptic disease occurs. Now the scientist of the Australia in 1977, he said that it is the disease which may caused by one of the commensal at one time. At one time it was commensal. That is Helicobacter pylori, for example. It was commensal at one time to the stomach. He said, no, it is not commensal. It is a bacteria, gram-negative, small bacteria, and it is the responsible agent for the acid peptic disease, responsible for the other disease of the stomach as well, like the lymphomas, like the carcinomas. It was not accepted by the Americans. And after many, many years, about 10 years of his research, proved in New York, in the Paris and in the United Kingdom afterwards, that yes, these are organisms, their mode of action was detected, and they are responsible for the acid peptic diseases and the related diseases he mentioned. And he got Nobel Prize after 12 years of his finding. What happened? The Americans never accept. After their heavy research, they found that he was correct. So that is why the microbes, at one time acid peptic disease was treated by the antacids and these things and that thing, or by the surgery. Now the surgery is very, very uncommon related with the acid peptic disease is concerned, because you will give the anti-H. pylori therapy and patient was cured forever by the anti-H. pylori therapy. And that is the actually magic. Alhamdulillah, we have the persons who have worked hard to detect it. I salute to them, all of them. Leading from our part of the world, the Arabic world, to the now, they invented it. मैंने दो चीज़ें याद रखी, तहकीक अगर आप कर सकते हैं और तनकीद बर्दाश्त कर सकते हैं तो आप तरक्की याफ्ता कौम होंगे। लेकिन अगर आप तहकीर करते हैं किसी की, तनकीद बर्दाश्त नहीं कर सकते और तहकीक करने को भी तैयार नहीं है तो आप एक जाहिल कौम हैं, अब आगे नहीं जा सकते और वहीं रुके रहेंगे और गलबा जो आएगा वो दूसरे लोगों का आप पे आता रहेगा। जज़ाकुमुल्लाह खैरन कसीरन कसीरा वमा अलैना इल्लल बलाग। अस्सलामु अलैकुम व रहमतुल्लाहि व बरकातुहू।